Okay, one last video in this autonomic nervous system lesson. In previous videos, we covered the effect of the autonomic nervous system upon the heart and upon the arterioles. This was excruciating, but it's all downhill from here because we covered all four receptors, and now we just have to talk about how they interact to cause effects in smooth muscle of the GI tract, and then later on the salivary and sweat glands. So in the GI tract, your muscarinic receptors, those associated with a parasympathetic response, increase motility. That should make some sense. You know, my parasympathetic tone is high when I'm sitting, maybe I've just had a nice meal, you know, and this is where you want to increase the motility in your GI tract and get that digestion going. On the other hand, the beta receptors and the alpha receptors, the beta receptor and the alpha receptors, they decrease that motility. When I'm running away from a bear, I really don't care how active my GI smooth muscle is. You know, my fight or flight, if I'm flying, which most of the time I'm going to run, you know, the motility of my GI tract is not what I want to waste my energy on, all right? So that's a decrease in response to really what are, for the most part, these sympathetic receptors, for the most part, okay? And so this should make some sense. Now let's talk about how it operates. All right, this is a nerve. I'm going to get to it in a second. This is a synapse onto GI smooth muscle. The neurotransmitter here is acetylcholine, okay? All right, and so this nerve will stimulate this muscle this muscle through the release of acetylcholine, all right? So we have a number of different receptors that I want to cover first that are on the smooth muscle membrane of the GI smooth muscle. The first is the M3 receptor. Remember that? We covered it with vascular smooth muscle. So based upon what this thing did in vascular smooth muscle, what do you think acetylcholine binding to the M3 receptor is going to do? Okay, what did it do in the vascular smooth muscle? It bound ACH. It activated GQ, it increased IP3, it increased calcium release, right? The increased calcium fed into our standard smooth muscle excitation contraction coupling pathway, calcium binding to calmodulin, activating myosin light chain kinase, phosphorylating myosin light chain, causing contraction. Without even seeing this, you could predict ACH binding to the M3 receptor will cause an increase in GI smooth muscle contraction, an increase in motility. And I'll be darned, that's what it does, okay? So this makes sense. If you hearken back to what we learned for vascular smooth muscle, you can apply that here and you can say, oh, it's basically the same thing. It is basically the same thing, you know? For all practical purposes, increased contraction through this response, that should make sense to you. High parasympathetic tone, high ACH. This is where we're digesting our food. We want high GI motility. So it should all fall together here. And basically, you could have predicted this knowing nothing else about it, okay, without me showing this. What about the beta-2 receptor? Also on the GI smooth muscle membrane, what do the beta receptors do? They activate GS, they activate adenylyl cyclase, they increase cyclic AMP, that increases pKa activity. pKa will have its targets in the cell. This is a smooth muscle cell. What do you think the target will likely be? Correct. Myosin light chain kinase, just like it was in vascular smooth muscle. Increased cyclic AMP, increased pKa, pKa phosphorylates myosin light chain kinase, that decreases myosin light chain kinase activity, causes you to have more myosin light chain and less phosphorylated myosin light chain, and that gives you relaxation, decreased GI motility. That should make sense. High sympathetic tone, I really don't care about how fast I'm digesting food. So I'm decreasing my GI motility in response to norepinephrine binding here. Very predictable, okay? Now, having said that, there are things that you just have to memorize, Okay? One of them is the effect of the alpha-1 receptor in this GI smooth muscle. I hate the word memorized, but in this case, I don't know any other way to do it. What the alpha-1 does is it activates a potassium channel that causes potassium to leave the cell. It makes the inside of the membrane more negative, so you get a decrease in membrane voltage, and that will decrease opening of voltage-gated calcium channels, and you get a decrease in calcium. Okay? Sorry, you're just going to have to remember that one. Now we have one more receptor. It is the alpha-2 receptor. The alpha-2 receptor is actually located in the presynaptic membrane, which is the neuron here. Okay? So what it does, it acts just like it does in a vascular smooth muscle. Decreased adenylyl cyclase activity, decreased cyclic AMP, decreased pKa in the nerve. Decreased pKa in the nerve, well, now you ask yourself, what are the targets? Well, as you will learn, I know you can't read this text, and it is not important for you to be able to read it. We will go through it at a later lesson, okay? But what you should know is that the release of neurotransmitter here is dependent upon calcium influx through a voltage-gated calcium channel, okay? And so, if you decrease the phosphorylation of this calcium channel, you decrease calcium influx through that channel because you decrease its opening. And that will decrease calcium here, and that will decrease acetylcholine release, and that will decrease activation of M3 receptors and cause relaxation. Okay? All right. So that's the way this works, and that should not be unfamiliar to you. This is what happened in cardiac myocytes. The even-numbered muscarinics and the even-numbered alphas act the same way. Decreased cyclic AMP, decreased pKa. In the case of the muscarinic, that decreased phosphorylation of L-type calcium channels, which decreased calcium influx. The same thing is happening here. We're decreasing phosphorylation of N-type calcium channels, voltage-gated calcium channels. That's causing a decrease in calcium influx, and everything flows from there. Decrease neurotransmitter release, decrease GI smooth muscle activity, because we have less ACH. All right? Okay. Finally, we get to go through the glands. The salivary glands and the sweat glands, okay? These act pretty much the same way. The difference, they both respond to acetylcholine. The difference is the sweat glands, the acetylcholine comes from sympathetic nerve endings. And in the salivary glands, the acetylcholine comes from parasympathetic nerve endings. So this is typical parasympathetic release. This is sympathetic cholinergic activation. Okay? In both cases, you are stimulating secretion. So let's see how it works. We have our M3 receptor again here. That is causing, as with all M3 receptors, all odd-numbered muscarinics, all odd-numbered alphas act through phospholipase C to increase IP3. Increased IP3 causes increased calcium release from the endoplasmic reticulum. And in this case, the target for calcium is chloride channels and potassium channels, both of which are activated by calcium. Calcium literally binds to them and causes them to open. Calcium and potassium go from the inside of the cell to the lumen of the gland. This is the basolateral side, this is the apical side, this is the lumen of the gland. So you have more ions accumulating in the lumen. That means the osmolarity is going up. If the osmolarity goes up in the lumen, that means more water is going to follow and go into the lumen. So more potassium and chloride in the lumen, water follows because this is increasing the osmolarity. And you get an increase in secretion. More water, more secretions. All right? So that's generally how both of these glands work. The only difference is the ACH comes from the parasympathetic nervous system for your salivary glands, should make sense. I'm digesting food, I'm eating, I'm relaxed, high parasympathetic tone. It comes from the sympathetic nervous system when we're talking about sweat glands, and that should make sense. I'm sweating, you know, I'm exercising, my sympathetic tone is high. So we have acetylcholine released from the sympathetic nerve endings. We have sympathetic cholinergic activation of M3 receptors, and we have more secretions, more sweat. All right? That's all I got. That's the end of our video. That is the end of our lesson. Okay, having gone through that lesson, here's what I'd like for you to do. I'd like you to fire up this ANS workshop program that I've supplied you with, okay? And I will tell you that this is basically a review of the same systems that I just went over in this series of videos and in the notes, okay? What I'd like you to do is to do problem 1 and problem 2. And these are problems associated with the autonomic nervous system. You will be walked through the answers, and I have supplied you with the answers, but I'd like for you to try to do the problems before you look at the answers, because I'd like for you to try to apply what I've taught you when I went over all of this to a couple of problems. You don't really have to go through these if you watched my videos. It's a nice review, but what's in my notes is basically word for word what's in this program. Okay, but these two I'd like for you to do, and then you can look at the answers and carry them away and use the answer sheet to study for your quizzes and your exams. All right? Thank you for your attention, and I'll see you guys in the rooms.